we're going to jump right into our panel on external control cohorts for clinical trials. The panel will be moderated by Ashley Margolis, who is the director of the Brain Tumor Center and Neuro-Oncology program at the Children's Hospital of Los Angeles. Joining the panel will be also Dr. Lindsay Kilburn, who is the director of clinical research and developmental therapeutics within the Brain Tumor Institute at Children's National. Dr. Annette Molinaro, who is a professor of neurological surgery at UCSF and is a biostatistician with extensive experience in clinical trial design. Ms. Amanda Haddock, president and co-founder of the Dragon Master Foundation and member of CBTN's Executive Council. And Adam Resnick, who's the director and co-director of the Center for Data-Driven Discovery in Biomedicine at CHOP, as well as a bunch of other things. And I neglected to say so before, but please allow me to do so now. The delicious lunch we all enjoyed was courtesy of the Dragon Master Foundation. Thank you, Amanda. And without further ado, Ashley, if you wouldn't mind starting us off.
Good afternoon, everyone. So just a little bit of housekeeping, the format of the panel. So I'll give a little bit of background about why we're even having this conversation, and then ask our experts to comment on various aspects of this idea of using external control cohorts to design clinical trials. And then we really at the end want to open this up. We want to be provocative. We want to hear from you all kind of your thoughts, your concerns, and then try to, if possible, come to some sort of consensus around how different people in the community feel about this idea. So I'm going to start, and I'm going to use my peripheral brain here to give us a little bit of background on the topic. And I should mention that Mike Prados was supposed to be the one moderating this session, and he's not here with us, so I'm going to try really hard to channel my inner Mike Prados and be provocative. So one of the biggest challenges that we face as a community is how to effectively and efficiently conduct clinical trials for children with brain tumors. And if we consider a disease like ATRT, for example, where there are only 50 children diagnosed in the United States every year, we think about the largest prospective clinical trial ever conducted in this country for those patients. That clinical trial enrolled 65 patients. It took 6 years to enroll and another 6 years to analyze and publish the data. During those 12 years, we learned a lot about this disease from a biologic standpoint, a molecular standpoint. We learned that there are 3 different molecular subtypes of ATRT. But because the trial was designed before we had any of this knowledge, it obviously was not powered to allow us to ask these new and important questions that we as a community wanted to ask. When we realized there are these subgroups, we wanted to say, okay, explain to us, which subgroup is going to do better? What are the different types of therapies that we might be able to harness to treat these different types of tumors? And we simply couldn't ask those questions because the biology outpaced the clinical trial. At the end of the study, we were left with essentially more questions than answers. And so now, 12 years later, now it's going on 14 years later, we're still stuck with a survival rate around 50% and not a whole lot of guidance as to what do we do for the next iteration of this clinical trial for this patient group. And we've heard a lot today about subgrouping tumors. And as we continue to learn more and more about molecular drivers and biologic differences in tumors, we continue to subdivide pediatric CNS tumors into narrower and narrower subgroups. And I like Sarah's kind of thought process around these splitters and the groupers. And splitting is good, right? It's good. It allows us to have more homogeneous patient populations, which is going to be very helpful when we're talking about clinical trials using molecularly targeted therapy. It's good to have homogeneous groups. However, it poses a whole other set of problems. which is now you've taken this really rare group of tumors and you've split it into rarer and rarer and rarer groups. And so your numbers for enrollment on clinical trials are going to go down. How do we get around this? How do we design a clinical trial for a disease that now we're talking about, instead of maybe 50 patients a year, occurs in less than 25 patients a year? How do we assess the efficacy for therapies for newly described tumors, right? There's new tumors now in the 2021 WHO. So what are we going to use for comparisons, for efficacy, for outcomes data? And also, what do we do with the other data? So again, I'm going to reference Sarah, but I love this, the tumor formerly known as peanut, right? So we have these outcome data for peanut. What do we do now that that tumor, is retired? It's a retired tumor, but are those data lost? Like, what do we do with these data? So I think the question we need to think about here is, how do we conduct trials more efficiently so that we can stay on pace with biologic discoveries? And as you've seen today, we're making biologic discoveries at a phenomenal pace, and we need to find a way for these discoveries to translate into clinical care at a faster pace than exists in our current framework. We also need to address patient and family desires. We're caring for a very informed and connected patient population, thanks to many of you in the audience. And they understand what treatment options are being offered to them. They understand randomization. They understand outcome data for what we would consider, quote, standard of care therapies. And we need to design clinical trials that they're excited about and that they're going to view as valuable, or else they're simply not going to enroll. Is it reasonable to randomize patients? Is it reasonable or even ethical to randomize patients to standard of care therapies that we know are not curative? I don't know. We need to think about that. And as we develop these comprehensive data sets that we've been talking about today, with patient level data that includes demographics, treatment data, outcome data, imaging, molecular data, we need to figure out how we're going to integrate these powerful resources into our future clinical trial design. So with that, I'm going to ask Lindsay if you could speak kind of to this topic from your perspective as a clinical researcher.
Good morning, or good afternoon, I mean, sorry. The day's gotten away from me. So, I think to kind of expand on that a little bit, as we think about some of the earlier phase clinical trials, phase 1 trials, the primary outcome for that historically is to look at toxicity, to determine the best dose, and to think preliminarily about finding efficacy signals. These trials are historically conducted in patients with either recurrent disease, where they may have received a variety of different therapies, and may have received, and often kind of various tumor types are combined in a single trial. And so trying to identify a signal there where your patients may have all 7 different diagnoses kind of in any particular cohort of patients, and trying to kind of then inform where to move these agents forward is very challenging. That has, similar to kind of how clinical trials have evolved and how the biology and understanding have evolved, has become a little bit different sometimes in the context of molecularly targeted agents, where we may be thinking not even just about a particular diagnosis, but more about the target. And there's been some recent examples of drugs that have received approval, kind of agnostic of pathology or diagnosis, but really more based on a particular target. When does that make sense to think more about a target more broadly versus thinking about, a trial for patients with DIPG or DMG versus medulloblastoma, and kind of how do we design the trials to kind of make sure that we're not missing signals in patient populations based on what we think we know, I think is really a current challenge. There are many examples of situations in past clinical trials, and I think we heard a little bit about ONC201 earlier being an example where there was a signal in a particular patient group. We thought a drug worked in a certain way. We've since realized we think it may work very differently. And I think there's other kinase inhibitors and things where we thought a particular target was the primary mode of action of a drug. And so if we think about a very specific patient population for a specific target, we might actually miss kind of other targets where that drug actually might be very effective, or other diseases. And so as we've moved and evolved, the kind of selecting the patient populations to study drugs in becomes very difficult. And yet looking at those efficacy signals and figuring out, when we are trying to look at preliminary efficacy, what are we comparing to? As Ashley mentioned, for many, many years, for example, children with DIPG or DMG are one of the groups of patients, because of the very, very difficult outcome and the fact that we've made so little progress there, where patients are enrolled at initial diagnosis in these phase 1 studies where we may not even know the perfect dose of a drug, and we certainly may not have efficacy information. And for many, many years, we could say that the outcome had not changed at all, and that we could kind of layer those outcomes, on top of each other. But we have thankfully seen some small shifts in the outcome for children with those diseases. And so we certainly don't want to go back and compare to kind of a therapy which we know isn't effective, but how do we identify the drugs or the therapies that are actually making a difference in that patient population? Kind of how do we create more contemporary controls, I think is a real challenge, both for studies like that, but also as we think about kind of how to interpret data from phase 1s, and how do we know that there's a signal there. Another big challenge in our early phase clinical trials is, what is it that we're looking at? Obviously, toxicity is straightforward. We have definitions for kind of what's intolerable, but to be honest, some of those are actually, based on historical types of therapies, and may not actually be intolerable. And we may throw away drugs because we don't escalate them well enough in the context of kind of current or a particular diagnosis where we might tolerate some toxicity beyond kind of what some of those standards are. But I think the other challenge, and we heard some this morning about kind of how do we assess response? We have very high bars for assessing response in clinical trials, and that's often based on imaging. We've heard a little bit about some of the adult imaging characteristics that that's based on, whereas now we know that those may not be applicable in pediatrics. And or do you require a 50% response in a disease where we almost never see that type of a response, but may actually see real clinical benefit? So I think that we, for many years, we're kind of designed these trials in a very prescriptive way. And there's been some really exciting advances in kind of accelerating and speeding up clinical trials. But we also have to really challenge ourselves to kind of really think about what we're trying to answer, what questions we're trying to answer, what restrictions we need to put on those trials. We often exclude, various therapies, and we don't do a good job of kind of combining therapies because we want to kind of be as straightforward as possible in terms of limiting kind of the variables, but that doesn't necessarily provide us kind of with real world data. And we may be comparing it to data and to other trials, which are therefore very dissimilar in terms of kind of the other confounding factors. So I guess.
Thank you so much, Lindsay. Maybe you can pass the mic to Annette. And Annette, I know you've thought a lot about this topic and worked extensively in this space, but can you just walk us through what is meant by external controls, external control cohorts?
Sure. So I'm Annette Molinaro. I'm a statistician at UCSF and worked with Sabina on the PNOC trials. So when we're talking about external control data, we're talking about data that we're taking from somewhere else. So patient data that is coming from a current registry or it can be a historic registry, previous trials that have been run. It can be from EHRs, different places. So we think of them either being contemporary, so the data is being collected right now, and the advantage there is that there's a similar standard of care, if there is a standard of care, or at least the imaging is similar, it's a similar timeline for those things. But then a lot of times we have to depend on the retrospective, the historic data. And in that case, the standard of care, if there is one, is not the same necessarily, and the timeline is slightly different. So those things need to be accounted for. But when we think about it, the reasons we're doing it are some of the reasons we laid out already, have been laid out already. There's a lack of a standard of care on therapy. There's low incidence, and there's this ever-evolving understanding of the tumor biology and molecular characterization, which are 3 prime reasons to use it in pediatric brain cancers, particularly brain. So we can have this contemporary or non-contemporary, and just depending on when it's collected. But the importance there is then what can you ask with that? And how can you ask it? So if we're interested in doing comparative treatment effect, so we want to compare a treatment arm to a control arm, obviously having the contemporary arm is going to be, or external controls is going to be best. And that would have to be through a current registry. Then, but then we can either in that situation take a trial and augment the control arm, or we can form a standalone control arm. So if we think about having just a 2-arm trial, so we have a treatment and a control. And what's nice, if we think about the contemporary, and we're allowed, and we have the possibility of randomizing, right? In statistics, the reason, and in clinical trials, the reason we use randomization and blinding is to alleviate bias and confounding. So selection bias, physician bias, patient bias, those types of things, but also the confounding due to age or location, SES type things like that. And so if randomization is possible, it's always a good thing, then you could start randomizing if you had a concurrent control arm, external control that you were augmenting your control arm with, you could start randomizing maybe 2-to-1 or 3-to-1, 2 in the treatment arm to 1 in the control arm. And after a certain number of patients were collected, then you could compare your current control arm to the external control arm that you have sitting in another database somewhere. And if it's similar, then maybe what you do is you change your randomization allotment to 9-to-1. So now you're putting 9 in the treatment arm and 1 in the control arm. And then you're going to use that external control arm to augment your control arm. So that's a fantastic use and way to decrease the number of patients that are going into a control arm that's either standard of care or, as we're talking about, lack of standard of care. So there's those situations. Then there's also, if we just use a standalone external control arm, so randomization isn't possible. So we have this, we have a treatment arm, it's a single arm study, and we're going to compare it to an external control arm. In that situation, if doing very simple power calculations on if we just compared it to a historical rate for, let's say, progression-free survival or overall survival, if we had a historical control rate, we can reduce the sample size by about 25%. So the number of patients needed by about 25% by using an external control arm with a good number of patients, 200 or 300 patients in that external control arm. So having that data available is exceptionally important and could really help us speed trials up. The important thing, though, is especially when randomization isn't available, is that you have to have meticulously detailed and annotated data. And that is important for all the reasons we've just listed, but mostly I would say for this idea that we keep on learning more. And we don't know what we're going to find out in the next few years, but it may be a new molecular marker. And so having whole genome sequencing or some of those things will be incredibly important now on these patients so that we can use them for the control arms. Because if you can't do randomization and you have to show, and this is a holdup that we have with the FDA in some adult trials, show that your external control arm is very similar to either your treatment arm in terms of clinical characteristics and things like that, you have to have really fine detailed data in order to show that comparison. And so, that's goes on to future things with databasing and things, but I would say those are some of them.
That's a great segue. So, Adam, maybe you can kind of help us think about understanding that we're going to need these incredibly detailed data sets. How do we bridge the gap from where we are now to where we need to be?
Yeah, so one of the things we're seeing is, and I think this is something that's hard to communicate, is if you take the CBTN data set as an example, is, we begin with, a predefined CRF that Angela defined at one point in time. We collect specimens at a particular point in time, but that data is not static, right? So we begin to then do large-scale molecular characterization, and something that was written in as a peanut, we now know is not a peanut. So we then backfill and inform that data. Likewise, as we essentially recognize that a clinical feature that may not have been included in the CRF initially, we can, right, deploy our army of coordinators to go get that data, right, from the EHR. So the part that I'm really sort of excited about is asking the question of, as we create these foundational layers of data, it may be that in one particular histology or setting, you'll need other data or other variables that are not present, or you'll need to generate, a molecular feature, right, that will subdivide your tumor into a very fine cohort. But really the guidance, that we need to impart and define is how do we essentially maintain that dynamic framework of adding asynchronously to the data to align that data with the prospective opportunities, and not think of retrospective data as static data set that are fixed in time, because they're not, right? None of the tumors that came into CBTN as peanut will be peanut, right, the minute we have sequenced them. So really thinking about data as dynamic. And then likewise, on a perspective framework, some of the tools and resources are creating the opportunities for, instead of sort of fixed in time data, just like on the clinical trial side, we have data streams of real world data. And I think creating parity of data streams between clinical trial perspective, treatment arms and real world data as data stream creates a framework of dynamic exchange between the clinical trialist and the real world data setting. I think those are some of the opportunities that we see emerging in the data landscape that we have not yet harnessed to fruition.
Great, thank you so much. And Greg, we'd welcome your input from the NCI and maybe some regulatory perspective if you can give us any.
And just to be clear, Greg, I think everybody here thinks that you've solved it. Just to be clear.
That I've solved this?
Yeah.
Yeah, why are we talking about this?
Yeah.
It's not a problem at all. Well, from a regulatory perspective, well, first of all, I think the question about clinical trials and efficiency of clinical trials. I like to think of clinical investigation in cancer is really sort of covers a very broad waterfront. So, what might be an efficient trial in a newly diagnosed population where you're trying to establish standard of care may be different than a clinical trial where you're just evaluating a signal of activity of an investigational agent. So I think first you have to really question what is the objective, or what are the objectives of the clinical trial. And what is the scientific evidence base on which a trial is in fact based? What rationale really supports human experimentation? But to answer your question, I think specifically about external controls and the potential regulatory use of external controls in drug approvals. I think that the FDA recognizes and clearly in pediatric oncology that randomized trials are impossible in many situations. Not in all situations, but in many situations. And there has been, I think, a real move to look at ways to use external controls, either historical data from registries, although that's probably the least useful because temporality is a big issue. And maybe the specific standard of care for that disease hasn't changed, but other things have, whether there's blood product support, antibiotics, other supportive care measures. And then the other is, how that data was collected, by whom, how it was managed, how it was curated, what's the provenance of the data, and is it auditable? Can you audit? Because there's no external control data that's going to be evaluated by the FDA unless it's patient level. So someone has to see it, and that creates a big problem because some data from clinical trials, there's no opportunity for that data to be made accessible to a pharmaceutical sponsor who wants to come in and get something approved. And that's why you really need to think about these things prospectively. And the whole issue of making sure that patients in the external control group are comparable brings up the issue of propensity score matching. And that again is something that needs to be thought of prospectively, not after a study is done and you find out that in a specific subset of patients that you enrolled on the study that the outcome was good, and then you're going to go looking for a control group. That's not how it works. You really have to identify what these specific indicators, prognostic indicators, disease characteristics, biological characteristics, sequencing results. You have to identify them first and say, this is how you're going to select your historical control when it comes time to do the analyses. So I was mentioning to Lindsay earlier that, we currently have a number of applications in-house for drugs in pediatric oncology, for pediatric oncology indications that are the results of single-arm trials using historical control. So, this is moving, but there's a right way to do it and a not right way to do it. I won't say it's wrong, but I think, my advice is speak to the regulatory agencies as early as possible. And if this is a study that's going to be done internationally, which most of these are, because, we're talking about small numbers of patients, then talking to people at the EMA and the FDA at the same time, early.
Thank you. Those are excellent points. And then Amanda, from your perspective, and perhaps on behalf of patients and families, can you give us your thoughts on this topic?
So I'm going to try. I can definitely give you my thoughts. Speaking for a community of very distinct individuals is always difficult, right? So I'll try to give some overarching things that I think most patient families can agree with. But first, I think I've been listening to all of you and things that you've said, and is really hard for me not to like interrupt in the moment and go, oh, this, because I'm going to try to remember the different things that were said. But one thing that I think that's important to note is that researchers and reviewers have come up in this academic system that I don't think serves the current clinical environment very well. And there are really some systemic changes that we need to make if we're going to be responsive to these patient families. And we need to be, right? Because the successes that she was mentioning earlier in those clinical trials and those people that are enrolling because they know they're not going to be part of that control arm. And like, I hate doing stuff like this, but like really, if everybody in the room just stopped for a moment and thought about it and thought, this is my child. Am I putting my child on the control arm? Or am I going to go to another country where I know I can get that drug? Anyone within their means is going to put their kid on a drug that's going to give them a chance, because the standard of care isn't curative for any of these kids. And as a parent, you want to do everything you can, number one, to give your child the best shot at a quality life. But number two, just to know that you did everything that you could do. And so asking them to enroll a child on a control arm just for the scientific principle isn't ethical. And I believe that because we don't, it's not like a diabetes drug, right? Like we know that these children are dying. We know that there might be an outlier that lives 2 years, right? But in the 12 years that we had that ATRT study open, how many kids died while we were waiting, right? They don't have the time. This is not a population that has the time to wait for those kinds of things. And I think that with patients especially in the brain tumor environment, this is their care, right? Like somebody in the room would probably know, but it's like 85, 90% of kids go on clinical trials. I don't think we can consider that something that they're doing just for research anymore. That absolutely has to be considered their line of care. And if so, what can we do to make that more useful, right, for the clinical trial process? Because I think Sabina mentioned earlier, like they're going to talk about it, right? The days of going, well, we're going to have this study and we're going to control the environment that this study happens, like that is not today's world. And we're never going back there, right? Not if you want patients to participate, they want to know, but we need to be more involved in that dialogue with them. We need to help them, understand that just because the first patient does well or doesn't do well, that's not indicative of the trial, right? We've got to better educate the participants so that they can be participants, right? And not just subjects. And I think that's a big shift that's really difficult for everybody to grapple with. But the only way we're going to be able to do that is through really open communication and helping everyone be as educated as they can be in the process so that we are all working on the same team for the same goals.
Are we allowed to make comments? Are we allowed to make comments? So Amanda, I really appreciate your comment and two things that came to mind. So one, Greg, I really loved your very concrete guidance because I think part of us feels that there are things we could do better to set the stage for external control. So for example, auditability, that's a technical solution space, right? That's not a theoretical context. Like we can actually make data auditable, right? We can, create thoroughfares for pharma, FDA, or others to dig into the EHR as deeply, as necessary. So I think those are like very tangible steps, right, that we as a community can take around infrastructure or the setup of the framework. And I think that's some of the guidance that I think, we as a community are looking for. The other part, Amanda, that you touched on, and I just wanted to sort of elevate it, in these rare populations, because of the communication across the community, you inadvertently create disparities of care based on socioeconomic factors, where you may inadvertently, right, position certain socioeconomic settings as those who are participating in trials that others would not. And I think that's a high risk that we actually don't talk about as often as we should. And we shouldn't let that happen just because of a requirement in that framework. And we should have ways to measure that and be proactive towards that.
I think that's part of our siloing, you know. I think the risk of creating these inequities is because we do things the way we do them. And we really need to think about doing things differently. I mean, do we really need to reserve early phase studies for a select few institutions in this country? I mean, you know, I think I've always had a problem with that for 20 years. And even when I ran the Children's Cancer Group's phase 1 consortium, I mean, why only 20 institutions? I mean, you know, there's 200 institutions that may be able to do this just as well. So I think, and you know, that's part of the problem, I think.
To say thank you as well. But I think that kind of back to several of the points that have been raised is that question of like the curating the data well. And, you know, I think things to the point of that in a phase 1 clinical trial, the degree of kind of clinical trial and physician detail in review of that data that is entered at multiple levels is different, and even different than a phase 3 study where there's a lot of things that are kind of very standard. And I think we haven't historically held like our registries or even the clinical data associated with, you know, some of the tissue, you know, kind of annotation to those same standards. And I think that is one of the challenges, you know, that I think we're tasked with. And families want to, you know, Sabina mentioned, often families want us to be sharing that data across platforms. Many of our patients may enroll in more than one phase 1 clinical trial and maybe in separate consortia where that data is not shared, and yet could serve potentially as, you know, external controls for a different study, or the patient could serve as their own control. You know, they may be on one clinical trial much longer than another, which actually is potentially a signal, you know, that we're not capturing because those might be in different consortia. So figuring out the way to harmonize things and to share that data across, I think really would guide, you know, might better guide some of our clinical trials. And to the point of kind of access, you know, I think, and equity, I think there have been some real advancements in the age of post-COVID, which had allowed us to do things and to bring trials closer to patients in some situations, but some of that has kind of come back, unfortunately, recently. And so I think we need to also figure out what are the ways to bring access to more patients. I do think there are aspects to early phase clinical trials which may not be well served in smaller institutions if there isn't kind of expertise or kind of or I think there are some expertise that come with some of those early clinical trials, which are important, but there are ways to kind of get that out into the communities. So
you can build expertise, you can build capacity. I mean, we talk about building capacity all the time, but I don't think we do it as effectively as we can. You know, and I think another model that maybe could be considered, particularly in the early phase setting with the multiply relapsed patients for whom, you know, there is probably no curative therapy, are, you know, platform studies where there are multiple, so no one gets randomized to standard of care or worthless standard of care, but you might be randomized to a different agents, and you know, if we really have the equipoise that we're supposed to have as clinical investigators, no one should know which one of those is better than the other. So, you know, that's another approach that we could use, but I don't think we use it as adequately as we could.
Hi, we have a question from online, so if you guys will let me read it. From Mike Prados, I would like the panelists to distinguish a trial that has quote-unquote regulatory framework versus a clinical trial design that may still create a new treatment option. FDA and other pharmaceutical companies dependent on FDA regulations may hinder development of practice-changing therapies until we get a perfect method for collecting patient-level data across consortium academic groups.
I think that's for you, Greg.
I didn't hear the whole question. But I heard collecting data, so I thought it was for you.
Would you like me to repeat that?
I'm sorry.
Would you like me to repeat that?
Yeah, good, sorry.
Okay, sorry. I would like the panelists to distinguish a trial that has a quote-unquote regulatory framework versus a clinical trial design that may still create a new treatment option. FDA and other pharmaceutical companies dependent on FDA regulations may hinder development of practice-changing therapies until we get a perfect method for collecting patient-level data across consortium academic groups.
I'm going to turf it back to you because I think it is incumbent upon us to maybe not develop the perfect system, but better systems that we have right now, right?
So I think what we're finding is that it's technology will not supplant humans, right? But even within our network here, I think you mentioned it, this Lindsay, right? That when you're performing a clinical trial, the coordinator or the clinician will take a particular amount of care in putting in that data. That it's not clear if that same level of attention is being paid in a registry setting. But I think that's the opportunity we have is, because data can be dynamic, it's much cheaper to go back and apply that level of care to the data. It's much, we have our community of clinicians, coordinators, right? Cheaper both in human life and time than start anew. And so this notion that we can iterate on the quality of the data is really the part that I think is exciting. And it's going to require like blood, sweat, and tears plus technology. But I think it's worth it for these populations. And I think we as a community are willing to do that. And maybe that's what Mike is sort of pointing to is that, what are the opportunities around designing a trial based on that versus the idea that you have to start fresh from a very structured framework. And maybe it's a statistical method or really an iterative dialogue of how do we run that experiment with the FDA, as a framework.
I guess I would also add, I think what Mike's raising is that question of, there's a long history of clinical trials that we design as clinicians and researchers, which really kind of aren't thinking about what the regulatory pathway is. And honestly, most of our studies for many, many years were kind of designed with what we think is the best, question and the best way to advance our clinical care, is to come up with an answer which will hopefully help us define a new standard of care. And we honestly didn't really think about the regulatory framework around that. But I think as many of our agents have become more targeted and as, pharma, and we've partnered with them in very different ways, I think that pharma is thinking about those things. And so getting access to those agents is now requiring, and getting access to kind of newer agents to incorporate earlier into our clinical trials is requiring that we think as well about kind of what that pathway may be, or at least partner with them as they think about what that pathway may be. And so I think that's the struggle right now, is that we're kind of now bridging both of those aspects, whereas in the past we really were more trying to kind of think about things simply as what do we think the next best step is clinically, but we're having to kind of incorporate some things which may lead to a regulatory pathway in a different way than in the past.
And I think
it's critical to know upfront what's the objective of this study, and maybe it's only a possibility that this could lead to a regulatory approval, but if that's a possibility, then act on it upfront, and create the kind of rigor in that trial design and the collection and the management of data, so that should it be positive, then it could be used. And that's why I think it's really critical to speak early, to, there's 3 big parties here. Well, actually 4, because I think you have to include families, but regulators, industry, and investigators. So they all have to be at the table.
And I also think that Mike is pointing to the incentive structure. If a pharma is given the option of spending X dollars on a prospective trial that they're guaranteed will be, reviewed by the FDA versus spending that same resource on a retrospective cohort where it's not clear to them that there'll be a path forward, we can see the decision they're making, right? So I think what Mike is sort of pulling the thread on is, how do we align the incentive structure for the appropriate effort to be placed and for the experiment to be done, as opposed to defaulting to the norm in order to de-risk, the approval process for pharma, right? In ways that patients and communities, are currently not aligned with. And how do we align that opportunity with the RACE Act and the other requirements they have, to advance this?
Hello. You guys are talking about like clinical trials and they're very data-based, but if you look back at 20 years of clinical trials and the strategy that goes behind them, I mean, the end goal of better treatments has failed. I mean, there aren't better treatments. Do you think that under the current framework that clinical trials are done, specifically for rare pediatric tumors, the next 20 years would be any different? I mean, went through this whole genetic revolution, it's not leading to anything. Do the clinical trials process need to fundamentally change to allow more flexibility, more individual care to say, look, we're going to try this drug and it's not going to ruin a whole clinical trial. It doesn't have to be compassionate care because we needed to try something. Or do you think the framework now with new technologies, new data sharing might lead to something in the future?
I think we're all up here having this conversation because we recognize that we need to do things differently. And I think we're at a really great point to be able to do that. We've got so much biologic molecular data that we didn't have before. We were shuffling around old drugs for years and years, right? Trying to find new combinations or new orders of things. But we've had this dramatic expansion of biologic and molecular data. And to your point, how do we harness that? Like, what do we do with it now? And I think this is where we're also at a really great pivotal time where we have novel statistical designs and novel clinical trial designs. And I think the community is opening up to being more willing to accept novel clinical trial designs. So we're not talking about, let's do this phase 1. Okay, there was too much toxicity, maybe not really an efficacy signal, throw the drug out. Let's, spend 5 more years to write a new phase 1 for the same population. I mean, you saw Sabine talking about PNOC 22. We're having these new novel clinical trial designs to get to your point, which is we're not doing a good job, right? We need to do better. And I think we've got things like new statistical designs and novel clinical trial designs, and we've got this, huge wealth of genomic data that's helping guide us. And I think we're going to do better. But we all have to be willing to shift, right? And so we have to have conversations like this and be provocative and try to push the envelope in some respects.
And I guess I would add that I think there are actually many examples of clinical trials that have improved kind of the standard of care and have advanced where we are. But there is clearly a subset of diseases where we have made no progress in the clinical arena or made very little, unacceptably little progress. And I think that's what drives a lot of these conversations, is kind of focusing in that area. And that has pushed us to, I think, change the clinical trial. And I agree with kind of all the comments that are made. But I also do think that the models have really improved. Our ability to look at new agents and new ways kind of preclinically has really advanced, and our understanding of the biology and the way, many of the trials that we were doing for 10 or 20 years before were really based on assumptions that many of our pediatric tumors were similar to adult tumors, which is clearly not the case now. And so I think that makes us all hopeful that we really are at a place where we're on the edge, we hope, of kind of really making some advances clinically, but figuring out how to translate those advances that have happened so quickly in the last 10 years on the biology side to quickly translate them into hopefully successes on the clinical side is, I think, again, why we're all here. But I do think there are differences in where we are now than where we were 10 years ago when those clinical trials were written, both in terms of what we're building on, but also in terms of kind of the way we're thinking about how to push things forward more quickly.
Which is, I think, we don't have any industry partners up here, but we do have to give them a lot of credit. And we've had better relationships with industry partners, and they've been really willing to help us in a lot of situations and circumstances. And those relationships continue to foster. And I think that's going to be really helpful moving forward as well.
So one of the questions I wanted to get the opinion on from a regulatory point of view, right? I mean, we are discussing new trial designs, better genomic data, but I think really one of the biggest challenges we have is what is the endpoint, and what is the endpoint that will then lead to approval, right? We are discussing RANO, low grade, high grade, RAPNO, and in the end, if it doesn't meet the 50% cut, it will not be approved, although clinically, we all believe for certain drugs might be really beneficial. So trying to understand a little bit the disconnect what patients and then providers, and I think that goes back to your, the communication with these different stakeholders, like sort of the definition of what is an endpoint that needs to be met for regulatory approval in what disease space. And I've seen very little movement on that, I would say, or maybe there's been lots of discussion, but we haven't really solved that issue, right? How to define endpoints that really should lead to an approval, and love your thoughts on this, or comment from the other panelists, how we could really solve that. And especially love to hear from the parent-patient foundation perspective, right? If we believe something is beneficial for our children, how can we not give them access to that?
Well, that's sort of a separate discussion from external controls, but you're absolutely right. I mean, I don't think regulatory agencies they may have the final word, but they certainly don't have the final understanding of what is the right endpoint. I mean, traditionally, for full approval, or I shouldn't say full, but for traditional approval, regular approval, overall survival is been the endpoint that has been used. Clearly, in the trajectory of a child's life with cancer, there are other endpoints other than survival or the end of survival that should perhaps be included. And we're having discussions now with groups looking at functional endpoints, particularly in some diseases like low-grade glioma, whether functional endpoints like strength, mobility, obviously with optic low grades, visual improvements, neurocognitive improvements. They're all things that I think are on the table for discussion now, because the reason is establishing a sufficient clinical benefit, and I think in some diseases we just don't have a good feel for what is a sufficient clinical benefit. So we need the input of families and patients who are actually experiencing these. I mean, the whole patient-focused drug development program is because of these very issues, and input from clinical investigators as well.
I'd just like to add on that I think this is very much a conversation about external controls because the outcome really matters. And so if it's a subjective outcome, it becomes problematic when you then go to compare a treatment. And so it's very important to have your outcome objectively measured and well documented, and whatever that outcome may be, but yeah.
So I'm going to counter that a little bit. Because I think it's subjective because the endpoints that a clinician or that a researcher are looking for are frequently not the same endpoints that families are looking for. And survival, even if something is a similar survival time, it might be a friendlier medication for the child. And so I think sometimes things get held up because at the endpoint might be overall survival. And if, drug A is the standard of care and their overall survival is a year, and we're trying to get approval for drug B and the overall survival is a year, and they go, well, it's the same, but it's not the same, right? Quality of life can be very different from that. And it might not be something that's measurable. I mean, one of the things that bothers me a lot, and it didn't happen to my child, but it happens to a lot of kids, they are put on these courses of steroids. And not only does their course of life dramatically change because of that, but parents, this is a little sensitive, sorry, but parents are ending up burying a child that doesn't look like their child, right? And nobody's measuring stuff like that, right? It might be on some quality of life scale. Well, the child had these effects from steroids, blah, blah, blah. But at the end of the day, there's a lot of criteria that don't enter the processes, the evaluation processes. And if families are involved in those conversations all the way through, I think that it will dramatically change the way you look at those endpoints. And in really meaningful ways for the clinicians and researchers too. But I think we've really got to break that whole system and look at it, each individual element of it, and see where we can involve patient partners so that we do have a better dialogue around those endpoints.
I don't want to put any constraints on this conversation because it's really valuable. We have reached 5:30, so perhaps if we could take one more question and wrap it up, that'd be great.
Let me make one comment. I'll break the rule and then say, just because we talked about, you made a point of, other endpoints, but at the other endpoint, I think we have an opportunity, and we see this now, is the biological endpoint. And I just want to sort of highlight this iterative biology, right? Our capacity to measure signal is increasing. So we can go back to tissue, back to blood, liquid biopsy, measure variables that we didn't measure early on and start pairing them. So this idea that when you have, robust biorepository linked to clinical data, right, we can redesign what we measure, right, at those levels in ways that I think we haven't fully harnessed as an opportunity. And your last comment.
Just to build on what Amanda was saying for 1 second, I don't believe there is a clinical trial process that's been developed for children, right? I think what we used was developed for adults. So that's part of the reason why the journey is so horrific for children. But my question is, and maybe I'm wrong, but my vision has always been with respect to data and the clinical trial process, is that why can't we use that data as a clinical trial? Why can't we make assumptions and write algorithms and say, what does that tell us? That could expedite the whole process of a clinical trial.
Is that on? Yeah. You mean let the data lead?
Yes.
Yeah. I like that.
Okay.
Are we going to say something?
Actually, I'm going to say one thing. We've talked about this a couple of times, and I think it's, I think I'm going to leave us with something very challenging, but I think for those of us that are both clinical and clinical researchers, I think we've talked about a few things that have made me feel stronger about this today. I think we really need to steal back our medical records from billing to medical communication. And if we put good data in clinically, it'll be much more able to be used and having sat in the dark and done 1970s chart reviews, like it's not the technology that's really our biggest problem. It's us focusing on what's important, and some of that is going to be keeping the information from the get-go the right way, and I think we can do that together.
It's an excellent point upon which to end. Thank you very much. Thank you very much to our panel members. We really appreciate your expertise and insights.